So thank you all for taking the time. This is actually a very distilled version of a larger webinar that we've conducted to discuss ISO cell counting standards. So if there are questions about some of the things that I'm going to skim over, please feel free to email and I can follow up. But ultimately, everybody that's doing flow cytometry or really any cell-based work has encountered the need to count your cells. And it ultimately really is the most routine measurement in biomedical research, in academic research, in drug discovery, in translational medicine. And historically, it often has been treated very trivially, almost as if it's an afterthought. I just have to count my cells. I'm going to go plate them anyway. I'm going to go run them on flow anyway. But with the rise of immunotherapy, such as cell and gene therapy, where the medical intervention is actually a cell product, the importance of an appropriate cell counting method has really come to the forefront. And ultimately, it's not quite as simple as many people think. So cell counting in and of itself can be very challenging due to the range of biological sample types that you're dealing with. So for example, if we use cell and gene therapy as an example, if we're talking about CAR T-based immunotherapy, first you need to be able to actually count your apheresis samples. So these are very, very messy, heterogeneous samples from individual donors. So they have lots of platelets, residual red blood cells, things that can skew your counts. Then you actually have to do your transduction or your transfection to genetically engineer your cell therapy product. And there you're going to have to count and assess viability. When you're actually expanding those genetically engineered samples in culture, you have to count your sample there as well. Then when you're doing your potency assays to assess functionality of those genetically engineered cells, again, you have to assess the concentration and viability of those samples. And then when you're dealing with dosing and ultimately infusion, you also have to, again, count and assess the cell health and the viability of those samples. So throughout this entire workflow, regardless of what sort of biological research you're conducting, these types of counts are integral to the process. And particularly when the endpoint of an assay is the actual cell product, the quality of those samples, and as a result, the quality of the cell counting method is really critical. So a challenge with having an accurate or a best practice for cell counting is the fact that we don't have any ground truth reference for cells. When we're looking at cell counting methods, a lot of times we're not comparing the appropriate methodologies. So in reality, in a perfect world, we want a method that is precise, meaning replicate counts are very tight with each other. You have low CV, and you want them to be accurate, meaning the data you're collecting is actually representative of what your sample is. However, many times when you're looking at methods for cell counting, you may have something that's very precise, but it's not actually accurate. And because there isn't a universal reference material for cell counting because of the diversity of different cell samples, cell products, cell types, there's no such reference material. We can use beads as a calibration measurement, but it's not representative of the diversity of different cell samples. So FDA implemented regulatory measurements based on the 21st Century Cures Act, which basically defines that all of these cell and gene therapy products have to adhere to certain guidelines for the quality of these biological products. And as a result, it led to the publishing of ISO standards for cell counting. So there are part 1 and part 2. These are very comprehensive guidance. These are accessible for people if you want to kind of dig in. But these are available to now guide people in selecting an appropriate method to assess the quality of your cell products. Now, it's important to note that Nexcelom, we are one of the main contributors to the ISO cell counting standards series. The founder of our company, Dr. Jean Chu, she's one of the US technical advisors to ISO. We collaborate with NIST, which is the National Institutes of Standards and Technologies, to develop appropriate orthogonal methods and standards to actually assess image-based cell counts and viability measurements. We've co-published a variety of publications, so we're really in the thick of it in terms of fit for purpose cell counting methods. And that's really the main principle here that I want people to take away with, is that cell counting and viability, yes, it's part of your process. And yes, many people kind of diminish the importance of it, but it really needs to be fit for purpose. It's not one size fits all. So the method that you're using and the instrumentation that you'll ultimately use to assess cell concentration, viability, and even other sorts of cell health parameters needs to be determined by your entire experimental workflow, not just the instrument you happen to have on hand. And ultimately, using a multi-parameter approach, including orthogonal measurements, is going to be the most appropriate way to assess that. So when you're looking at the most appropriate method, your fit for purpose, you have to consider what type of cells you're working with, what sort of matrix are they in, or media, what method are you actually using? So there are so many different cell counting methods out there. Nexcelom, we make imaging systems that are based on bright field and fluorescence-based images and software to assess those. But there are, of course, manual methods, there are flow-based methods, there are impedance-based methods. And then ultimately you have to consider what your purpose is. So is this basic sciences research? Is this quality control? Is this in-process assessment? And all of these steps are going to have to define what your method, your most appropriate fit for purpose method is. So it has to be, you have to consider when you change a single parameter in that process, that could ultimately change your cell counting results. So say you historically worked with exclusively clean cell lines, so you always use trypan blue, but now you're switching to digested tissue samples. Trypan blue now is not necessarily the fit for purpose method for monitoring primary samples. So anytime you change something in that workflow, you have to revisit what your appropriate cell counting method is. So and one of the challenges that Nexcelom encounters is that many people want to compare one of our instruments to something they're currently using or another instrument that's out there to make a decision. And the challenge becomes, as you remember from the beginning, there are instruments that are very precise, but they are not accurate for the cell sample in question. And so the current mindset is often, well, let's directly compare method A, B, and C, and we want them all to match. And that's not appropriate, because if method A is precise but inaccurate, then methods B and C ideally should not correlate to that. They should be accurate. So because there's no ground truth reference for live cells, it's very difficult to use a previous established methodology as this benchmark. And so ultimately, that's really where the ISO cell counting standards have come into play, and including an orthogonal, unbiased methodology as, I want to say, a tiebreaker can help determine what a benchmark may be. So how do we actually select these appropriate cell counting methods? It's going to determine, we apply these ISO cell counting standards. That enables us to select the appropriate method, evaluate the quality of those methods, bridge between existing and new methods, because again, it isn't expected that they should match 100% of the time if one's appropriate and one's not appropriate, and ultimately improve your cell analysis quality. So for example, you have to consider your composition of your cell samples. So say we have clean cell lines and they have high viability, then, one method might be appropriate for that. But say we have a mixed sample or heterogeneous sample, or say our media has a different pH than biological pH, or say we have anucleated objects in our sample. So all of those things are going to affect what method you actually select and ultimately your results. So in this example, we have clean CHO cell line. very high viability. You can see that single trypan positive cell in the center. There are no visual particulates. It's very homogeneous. So in that situation, we may use trypan blue as a viable method. If we're looking at a similar sample of CHO cells, but with low viability, there have been published data that demonstrate that trypan becomes inherently less accurate when you drop below 80% viability, even in clean cell lines. So then the question becomes, okay, well, we want to measure live and dead cells. We want to look at this membrane permeability as a function of trypan blue, and we want to quantify our dim dead cells and our bright viable cells. So again, choosing the fit for purpose, you have to answer all of these questions. Now, if we move into this next example and we have a human leukopak sample, are we going to use trypan for this? Some people may, but there are other better fit for purpose methods to assess something like this. So a leukopak is going to have PBMCs, it's going to have residual red blood cells, it's also going to have platelets in there. So if we look at that, we have actually more accurate nucleated cell-based viability stains. So here we can use acridine orange and propydium iodide to assess viability. So here's a zoomed in image in the bright field of a leukopak or PBMC sample. We want to assess live and dead nucleated cells. So we want to remove red blood cells and platelets from skewing our counts. So here, when we look at these red arrows that are indicated, if we were counting them in bright field using trypan, we may include them. However, they are non-fluorescent because they're anucleated cells. We have some nucleated cells indicated in blue arrows here, and those are in fact fluorescent, so we can quantify those. We want to make sure that this fit for purpose method is not skewing our data. And so in this instance, for primary samples, the appropriate measurand is going to be acridine orange and propydium iodide. So if we actually use that, you can overlay that, and you can see if you were using bright field based methods, you would be overcounting by a substantial margin. And so the same method would be applied for all sorts of primary samples, including PBMCs, apheresis, tissue digest, BAL samples, other sorts of heterogeneous samples such as stem cells or hepatocytes, and also low viability samples because trypan blue becomes less reliable. So again, the cell counting system is going to depend on the assay that you're doing. So the instrument that you're going to select is going to have to be able to support the reagents and the methodology that you're utilizing and be able to assess things accurately. So even in the Nexcelom portfolio, we have instruments that are only suitable for certain cell types, say they're bright field only, but when you move into more complex samples, then you have more complex instrumentation. You want to select a cell counting method that's going to cover a broad dynamic range because, of course, the more times you dilute or manipulate a sample, that's going to induce additional sources of error there. And then, of course, if you have additional parameters, you want to consider these additional reagents and probes that you may also want to include. So other considerations, and I'm not going to get too much into this, but there are many, many parameters to consider when you're selecting an appropriate cell counting system, including what sort of regulation you need on that, sample volume, assay time, et cetera. And so ultimately, this entire workflow is going to dictate your cell counting because it is in fact a process. It's not a simple, endpoint just before you do your assay of interest. So it's important to really understand how you're sampling your samples, how you're diluting your samples, how you're treating your samples, and other sorts of things that may introduce error, such as settling time. So we've seen that even with cells that are in suspension, if you let them sit for a few minutes, it's going to affect and introduce additional sources of error. So those are additional parameters. You want to also be able to maximize how many samples you're counting or how many cells you're counting, and that's going to further improve precision. So we have a very comprehensive ISO cell counting metric that we use for evaluating cell counting methodologies. I'm not going to get into that at all today, but I'm happy to share that with you all. But ultimately, you should understand that anybody working with cells, you want a fit for purpose, not a one size fits all method, that it is a process in your cell based biology. And then you must factor those types of variables into your cell counting analysis. And I'd be happy to take any questions.
Great. Thank you very much, Andrea. In the interest of time, I'm going to go